Throughout human existence, man has sought to map the world around him. Now he has succeeded in mapping himself. Scientists have finally decoded the entire human genome, providing us with precise details of the 40,000 plus genes that define our species. Each of these genes is like a blueprint, which contains the plans for a particular protein. Every cell in our body has a full set of these blueprints, which it uses to build proteins, nanomachines that do the work in the human body. Rather than work directly from the master blueprint recorded in DNA, the cell runs off disposable copies written in a different material, RNA. The cell will use this disposable blueprint to make the protein, handing the instructions to machines called ribosomes. The ribosomes read the instructions and begin to make the protein it codes for. Afterwards, the instructions are recycled. LSBC has invented a technique where cells read blueprints we give them, so they can make proteins to order. This unique technology is called GeneWare. The first step is to prepare the work order. The disposable RNA blueprint is manufactured for the protein we want to build, for instance, a therapeutic drug. The blueprint is placed inside a virus, which is used as the delivery system to the cell. The virus is initially introduced into a few cells through abrasion of the cell's surface. Once inside, it hands off the RNA blueprint to be copied. The blueprints are passed directly to the ribosome machines, which read them and begin to produce large quantities of a particular protein. Since these instructions aren't incorporated into the cell's own DNA, the plants are not genetically modified. GeneWare is therefore not transmitted to future generations. The cell also copies the plans for the virus, which its own machinery begins to mass produce. The newly made virus are then dispatched to neighboring cells through interconnecting portals. In this way, the virus spreads through the host. Typically, this process is carried out in a plant, and every cell quickly receives the instructions to make the protein of choice. To collect the protein, the plants are simply harvested and processed, yielding large quantities of the protein with unprecedented speed, from days to weeks. Thus, for known genes and proteins, GeneWare can be used as a biomanufacturing tool, delivering protein products with extremely high efficiencies. GeneWare can also be used to discover the function of unknown genes. First, each gene or blueprint obtained from a cell sample is separated and copied into RNA. The RNA blueprints for each protein are introduced into different GeneWare viruses. Each virus, with its new blueprint message, is then introduced into host cells. When the messages are read, they will cause the host cells to gain, enhance, or even lose some natural function. By testing for change in function, and by identifying the blueprint messages that caused those changes, GeneWare can be used as a highly efficient tool in creating a catalog of gene function. Large-Scale Biology Corporation is assembling a collection of such catalogs into proprietary database libraries to empower the discovery of diagnostic products, novel protein targets for drug development, new genes for improved industrial and agricultural products, and the identification of new vaccines. In bypassing DNA and focusing on disposable messages using RNA, GeneWare technology enables LSBC to satisfy two important areas in the life sciences: safe, efficient, scalable biomanufacturing and high-throughput, high-speed functional genomics. GeneWare, the business advantage for genomics and biomanufacturing.